Good morning. Right now, somewhere in your body, a cell is dividing. It's been doing that since you were a single fertilized egg, 30 trillion times. The cell cycle is one of those topics that sounds like a list to memorize G1,
Mitosis.
S, G2. Done, but the exam doesn't ask you to list, it asks you to explain it. Why do chromosomes condense, what actually controls when a cell divides, and what happens when that control breaks. So, in this lesson, we're going to work through all of it. I've got two things to help me today. First of all, I've got one of my slide decks. This has been pieced together over the years, so apologies for any intellectual property right infringements, but most of the pictures have come from BioNinja, which is a fantastic resource if you want to check it out. And then we'll look at my little HTML summary you can find the link at the bottom and download it. Now, why do cells divide? What's the purpose? Why can't they just keep getting bigger? So the idea is we can link this back to surface area to volume ratio. Think of a bodybuilder. Think of, I actually talk about like that program with 600-pound people. I can't remember what it's called, or a hugely overweight person where they have to like take it out of the building with a crane. Now, I'm not being insensitive, but obviously you get very, very hot if you're that size. Why do you get very, very hot? Well, you've got a really poor surface area to volume ratio. It's really low. So you might have remembered this from earlier in the course, but the idea is you can't get rid of that heat. If heat's being generated in the center here, it's like so far for it to radiate and dissipate. So what happens is when the cell gets to its optimum size, there's like a trigger point where it's going to divide. Now, that division is called cytokinesis, and it looks different in animals and plant cells. And we need to know the difference between the two. A really nice way to put this together is in a table. You can easily construct a nice little comparison table. And that's a question that I would ask my students to do at the end of a class once we've done this. The main things is you've got this contractile ring with animal cells. You can see here microfilaments, and they are constricting, and then it's going to pinch in half. And it's pinching a cell membrane in half. So all they've got to worry about is the cell membrane in a eukaryotic animal cell. Whereas in a plant cell, we have a cell wall too. So one of the biggest difference is that the cell plate has to form, which is a cell wall made of cellulose, and it's deposited using vesicles containing the cellulose, and then it will construct a cell plate first. Now, we have some examples that have been put into the IB curriculum of unequal divisions. One of them would be in oogenesis. This is a creation of egg cells in humans or in mammals. And the reason for this unequal division, we end up with 4 cells at the end of meiosis, like 4 genetically different cells. And that's not incorrect, but when we're thinking about the formation of an ovum or an egg cell, we're only really ending up with 1 and a bunch of what we call polar bodies. So the idea of the formation of those polar bodies is so we have crossing over, random assortment, and we end up with genetically different eggs at the end. But the idea of the unequal division is so that we end up with a bigger cell that has a large volume of cytoplasm, and it's ready to receive the sperm cell. And the sperm is literally just the nucleus. Now, the other example would be in yeast. So yeast have been introduced into the syllabus when we make a comparison. We don't just compare animal, plant cells, we also compare yeast cells now as well. One of the big differences is in the cell wall being formed from chitin, whereas it's cellulose in a plant cell. And in bacterial cells, which are prokaryotes, it's peptidoglycan. And here you can see that they're dividing by a type of binary fusion, but it's not called binary fusion. This is called budding. And it's like a new one's budding off. Binary fusion is in prokaryotes, but it is the same. It's like cloning themselves, and it's budding off from the original parental cell, and we get a smaller daughter cell. Now, the bit that to focus on past this, so we've thought about, we'll go back to the outcomes. We've thought about describing why cells can't divide, and we've actually already explained the difference between plants and animal cells in cytokinesis. What I'd like to draw us attention to now is the steps. But before we think about the steps, now I just outline them. I always love a mnemonic. So pink mice are terrifying. Now, prophase, metaphase, anaphase, telophase. I had a student come up with one that was even funnier. It was like, I pull MILFs at the club. Slightly, in a gray area, almost inappropriate there. But what I always say to my students is, I'm going to give you a nice, cute one. But if you come up with a funny one that's maybe slightly rude, but it's like memorable to you, then you've got a more greater chance of actually memorizing that and remembering it for an exam. But I love a mnemonic because a lot of these words are very similar and it helps make sense of them when we're first getting to grips with the terminology. Now, the biggest thing with the terminology when we talk about cell division, mitosis or meiosis, is we want to think about what is a chromosome, what is a chromatid, sister chromatids. So it's all to do with whether or not they're joined. So here is one chromosome or one chromatid. At the end here, after condensing and it's been wrapped itself around histone proteins, now here they're joined, the DNA is copied, and we have one chromosome still, but we have two sister chromatids. But when they separate, we now have two chromosomes and they are a chromatid individually. So it's all to do whether they're joined. If they're joined, like maybe by a centromere at this point here, then they are now one chromosome and two sister chromatids. So it's really important, less in mitosis, but super important in meiosis to kind of get that terminology correct. We've got some other words. We've got a kinetochore, which is this protein that is forming the centromere. And it's just what's holding them together. And then when we go through the cell cycle, so here's a demonstration here. When the spindle fibers contract, so this is, we have our centromeres. This is a bit small. Let's see if I'll just make that a bit bigger like this. I'll get rid of that. And when the centromeres will release the, sorry, not the centromeres, got my words muddled there. I completely blanked on that word now. These guys here, what's this? What is this? See, teachers do it too. But the centrioles. So the centrioles, the little organelle that we often don't label when we think about a eukaryotic cell or a plant cell. They're not present in plant cells, but like in a eukaryotic cell, in the animal cells, then the centrioles are going to form a spindle fiber, which is a microfilament of protein. It will attach to the centromeres, and then there will be this contraction. And when it contracts, it pulls apart the sister chromatids, and then they become chromatids or chromosomes in their own right. They're then one chromatid still, and they're a chromosome. So the key part here, we've got this contraction of the spindle fiber. Now, I've got a nice little summary of this that I'll show you in a second on my HTML, which I'm calling a web sheet. But to quickly talk about the substages. Now, this is a cycle. We call it the cell cycle because we go round and round and round. Now, mitosis is one small part of this. You have 3 stages. Now, this is a HL detail, but we have growth, G1, DNA replication in S phase, and then growth in G2. Now, what we mean by growth as well is that the cell's getting bigger, the cytoplasmic volume's increasing, organelles are replicating, but also just normal stuff's happening, like protein synthesis. The cell's carrying out its function, be it like a pancreatic cell creating enzymes that it excretes, or something else. It's going to be carrying out protein synthesis for the role and the purpose that it's been differentiated for. So if you want to check out the little web sheet that I have, here's my website. Now, I'm building it out for teachers and students. And under resources, there is, down here, under IB, we have interactive lessons that you can scroll through. Now, these are IB and AP. But the cell cycle, now, the bit that I like in this, now you can read through this at your leisure. The idea is it's a nice little summary and it's something that works offline. So if you haven't got an internet connection, you're traveling, then you're able to have a little look at this then. Now this little scrolling here, this makes it come alive a little bit. So they get lined up along the center. And this is, this is metaphase where they've been lined up. And then as we go into anaphase, they get contracted and they get pulled to the poles. Now, what's triggering that? And if we look here, what we've got going on, you can see there's something called CDK, cyclin dependent kinase. Now, it's in a higher level there, and it's in its peak in mitosis. Now, we've got different cyclins. And that's dependent on which part of the cell cycle we're in. So in G1, we've got cyclin D. And then the one that peaks in S phase is obviously triggering DNA replication is cyclin E. And what the IB want you to remember here is that the cell cycle progresses due to peaking of cyclins. They were discovered by serendipity. A guy that was studying sea urchins noticed these protein complexes peaking at different points when he was observing cells at different parts of the cell cycle. And mitosis here is being controlled by cyclin B. Like serendipity is such a nice word, and it's really cool that this guy discovered that. He did that when he wasn't even looking for it. So it's really important, obviously, to look at all of your data and try and think about not just what your hypothesis is, but what else could be explaining what's going on. Now, at the end, I've got a nice little IB question that you can type in your answer to, and then you can reveal the mark scheme. So why don't you give that a go? And if you like the format of this video, or if you have any suggestions, please drop a little comment, share it. This is my second video. I'm developing the style that I think will work best, not just for my students, but for everyone. All right, thanks for listening, and I'll catch you next time.